What's up, AP Psych? Today we are going to talk about the eyeball. Wait, I'm going to take it really close. Eyeball. Ooh. Some people will find this fascinating. Some people hate the idea of talking about eyeballs, but either way, we got to know this stuff. So let's get to it. You can see there are a ton of learning targets for this topic. Of our five senses, six, seven senses that we're going to talk about in our sensation chunk here. This one's probably the most detailed. So put on your A game in terms of that note taking. Let's get to it. I actually suggest pausing me and filling in all of these parts of your eye. And then I will just add that the cornea is the transparent outer covering of the eye. Everything else we're going to get into throughout the lecture. So you don't need to define it in more than one place, but that one I only define right here. Um, I guess to take you on a quick tour, light from an object in the world will come through the cornea, which is why sometimes you might hear someone say that they scratch their cornea because it's the outer layer. But then it will go through the pupil, which is the hole surrounded by the iris, which is the muscle. So the pupil is not actually a black dot, it's actually a hole, and it just appears to be black. And then the iris is the muscle, but it's also the colored part. And then we have a lens beneath that, which then focuses the light on the back of the eyeball where we have the retina, which is kind of this whole layer right here. We're going to have a blind spot. That's kind of a fun one to talk about. And then the fovea is the area on the retina that has the best acuity. I'll talk about that, which means the best detail and color vision. And then we have an optic nerve, which travels the signal, that takes the signal um, through a process of transduction to the brain. And then we'll talk about where in the brain. But at this point, you should actually be able to tell me two or three parts of the brain that this optic nerve will eventually send the signal to. And if you really push yourself, I bet you could guess. Let's keep going and see if you're right. Okay, as I said, the pupil is just the hole that allows light in. The iris is the muscle that controls that pupil. That is the colorful part. And we know that the iris is actually the muscle that, well, the iris is making that pupil bigger or smaller. So the iris is the thing actually doing the moving, but we would talk about it and in terms of what's happening to the pupil. For light adaptation, like when it's really bright, like right now I've got this white screen flashing on my face. And so my pupil, I'm like trying to like check my eye out in the little bubble and also go close to my camera at the same time. My pupil is pretty tiny because I'm looking at a bright white light. But if I made it darker, let's see if I can do this. This is not going to work at all. Nope. If we were in a dark room and we could turn off or if we turned off the lights and we had a mirror, you could see your eye, your pupil dilate. When there's a lot of bright light, our pupil gets smaller to allow less light in and not be blasted. And then when it's dark, the pupil dilates. Dilates means get bigger to allow more light in to help us see better. Okay, pupil. There it is. All right. The lens's job is to focus light on the retina in a process called accommodation. What's funny is, and I know that this is a creature from SpongeBob, right? And every time I get yelled at by students for not knowing his name, but I just found this cute graphic and then did something with it. I don't remember his name, sorry, but it's fun for you because you probably do. Anyways, um, the lens focuses light on the retina through a process called accommodation. And what's interesting, you don't need to write this down, it's just kind of cool. When it comes through the lens, it actually ends up inverted and upside down, but then in the brain, we actually kind of write the image. Oh, kind of a weird, who knew sort of thing. Anyways, a distortion problem with the lens, alteration of this process is how we end up with nearsightedness or farsightedness, which I will get into in a little bit. Okay, on to the retina itself. This is the most important part of the eye anatomy that we need to know. This is the light sensitive inner surface of the eye. It contains rods and cones, which we will talk about much more on the next slide. And these are the photoreceptor cells for visual transduction. That is a very fancy phrase to explain things that we actually do already know. Photoreceptor cells means neurons, afferent neurons, like pew, 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 catching it with my dendrites on my receptor fingers of my dendrites. So it means they capture, this is the photoreceptor cells are your afferent neurons that capture the images from light and then change it into a brain signal, a neural signal. That's what transduction is. We learned that in the last lecture. So again, light comes through the eye to the retina, and then it's going to hit the rods and the cones and bipolar cells and ganglion cells. You need to write that down, but we don't need to know a lot about them, just that they exist. And I feel like I was going to say one more thing, but then ultimately it will go to the brain. We're going to put all of these in a nice little flow chart so it makes sense in a couple slides. The photoreceptor cells on the retina are rods and cones. You perhaps have heard of them before. Rods are sensitive to light, resulting in shapes and movement. So just with your rods, that's all you actually need to see shapes and to see things move. They really, to an extent, only show shades of gray. And they are only located, I like this diagram because they're located, it shows us on the periphery of our vision, on the periphery outer edge of the retina, just like we have our peripheral nervous system on the outer parts of our spine and our brain. It's like the outer edges. Cones are sensitive to color and detail. The center of the retina, the part I said on the very first slide, was like the most has the best visual acuity. That means it's the center of the retina is called the fovea. I feel like I lost my train of thought there. The center of the retina is the fovea. This is where cones cluster together. Looking back at this picture, you can see how they cluster together. And that's where we have the best visual acuity. So we have rods and cones, and they're basically in the shape of rods and cones on the back of the retina, specifically in the fovea, if we're talking about cones color. And then they connect to these bipolar cells and ganglion cells, which then will lead to the optic nerve. So as I just said, the retina leads to bipolar and ganglion cells, which form the optic nerve. So this is kind of a cool picture. This is the back of an eyeball. This is the fovea. Like I said, it has the strongest cluster of cones for detail and color. And then slightly to the side, behind, I guess it depends, like picture your eyeball being on like a cord. Or like, did you have one of those punching balloons that was like attached to like a rubber band growing up where you were like, you could just hit the balloon? It's kind of like that rubber band is like the optic nerve. So picture a ball on a string, and that's a string. Anyways, the blind spot, we have a blind spot because where this optic nerve leaves the retina to go into the brain, there are no receptor cells. There's no rods and cones. There's nothing else there. So we actually have a blind spot, a spot in our vision, and I'm going to do a cool demo in class that will demonstrate the fact that we actually all have a spot in our vision where we actually can't see anything. But you might say, well, that's not true. I see just fine. Well, what happens is your brain fills in what's missing in your visual field with nearby visual stimuli that, first of all, it just logically like says like right now I'm looking at my office wall, my home office wall. It's kind of a gray paint. And so instead of seeing like, I don't know, a black spot or something, my brain knows that the wall is gray and so it kind of fills it in. And one of the ways it knows that is from the support of the other eye and the fact that our eyes are constantly moving. So we don't really hold still in one spot long enough with our vision or with our partner eye for our brain to ever really register that we have a blind spot. But we will make the blind spot appear, see what I did there, in a demo in class. So as I said really fast at the beginning that we were going to put this all in a flow chart. But one more time, and this is another good place to pause me and fill this in. Wavelength of light enters the eyeball through the cornea, as we said, and the lens is involved here too, but less important. Light comes in through the pupil surrounded by the iris, and it hits the retina, which contains rods and cones. It's then transferred by bipolar and ganglion cells. to the optic nerve or an afferent neuron that leads to, this is the part that I was wondering if you could guess the brain parts, the sensory switchboard, that thalamus, which then sends the visual signals to the correct part of the brain, which is the visual cortex in the occipital lobe. This is quite the path that you have to memorize, but you do have to memorize it. If you think about it logically, it feels like more attainable, more doable. So light comes in, then we have some cells that process it, then we have the nerve that takes it to the switchboard, and then it lands in the correct spot in the brain. So if you actually kind of think through where the light is going and the signal is going, it's not so bad to memorize. Feel free to use the initials of some of these words to make a mnemonic device or a silly sentence if you think it will help you. Oh, and then star the retina because that's where transduction happens. So arguably this is the most important part of this process. That's where it goes from light into a brain signal. A light signal to a brain signal. That's probably the best way to say that. Speaking of light, light is the source of vision. So we see things because of light waves that enter our eye. Light waves are really a source of energy. Sometimes it's regarded as an electromagnetic spectrum. I actually don't know how important it is that you know that word, but definitely write it down. And then different light frequencies create different colors. So frequency is determined by the wavelength of a light. Like if you look at the purples here, we would call these short waves. The short waves are set blues, the medium waves are in the greens, and then the long waves, like it's more stretched out, those are the reds. They are measured in nanometers. I wouldn't expect you to have to memorize that, but it's another good thing to write down. Those are just kind of some supportive details. So you recognize it when you see it. You should not have to come up with that on your own. We will talk more about wavelengths in class. But one more detail I do want us to include in our notes is that intensity of light. So think about like the brightness on your phone or the brightness on your laptop, how you can turn it up and turn it down. That is the intensity of the light or the brightness of the light, and that is determined by amplitude. Like you can turn up and down the height of this wave by turning up and down your brightness. So brightness, amount of energy in a wave determined by amplitude. So this diagram is really good to show you amplitude meaning height, wavelength meaning how long does it take for a wave to complete itself from low to high and then back to low again to do it like a complete wave cycle. So wavelength, how long does it take to do a complete wave cycle? Amplitude, how high is that wave? Okay, now that we have down the concept, the anatomy of the eye, the concept of how light turns into visual signals in our eye, we're going to look at theories of color vision. Like, yes, we've got cones, but where do the colors come from? How does that work? Well, believe it or not, we actually have 6 million cones in each eyeball. So we have like 12 million cones in our bodies. And the trichromatic theory states that the retina contains only 3 types of cones, and they are sensitive to red, blue, and green. And these 6 million cones in each eyeball combine to allow us to see other colors. So we can see a full color spectrum because of certain cones being activated or inhibited at any given time. Some of our evidence of this is color blindness. When someone is colorblind, what that means, or color vision deficiency, which is kind of the fancy way of saying colorblind, a person can't tell the difference between certain colors. This can happen because either there's a weakness in cones or ganglion cells, or perhaps there was damage or a genetic deficiency. Most often, color blindness comes in or affects mostly reds and greens as a pair or blues and yellows as a pair. So in dichromatism, it means 2 of your 3 types of cones work. In monochromatism, only 1 type of cones work. And so you can see while this might be a normal apple for someone who is colorblind, it might appear like this, or this, depending on what sort of cones are activated. Perhaps some of you in class are colorblind and don't see the colors that the rest of us see on this screen. The opponent process theory is the second theory of how we see color vision. And this theory states that primary colors are not processed due to the activation of some cones and other cones, but rather they're processed in opposing pairs. And that ganglion cells in the retina, the ones that are attached to the cones, are either stimulated or activated, if you will, while others are inhibited. So it's a little bit like the first theory, only that these work in pairs of blue, yellow, red, green, and black, white. Instead of some just being on or off, it's more that when one is strong, the other is weak, and so on. I actually have a really cool demo to prove this, but I really can't do it on the screen. So the proof is going to be in something called an after image. If you think about it, when someone takes your picture with a flash, those spots you see are basically an after image. And we're going to do something with this picture in class, but for the moment, just get the notes down that after images are an image that remains after a certain stimulus is removed from your vision. A positive image would be something that looks normal, like a white light if we're talking about a flash on a camera. And then the negative image is a complementary or almost opposite, opposing opponent process theory, opposing image like bright yellow flipping to dark blue. Again, this is going to make a lot of sense when we do a demo in class. So just write it down and then it'll make a lot more sense later. Okay. Finally, last topic is disruptions to vision, like what can make vision go wrong. The most common disruptions are something that many of you might experience, and that is nearsightedness or farsightedness. If you are nearsighted, it literally means the things that are up close are clear and the things in back are fuzzy. If you are farsighted, it means the stuff up close is blurry and the far stuff is clear. What it means is the shape of your eye is a little bit different due to your lens, and a nearsighted eye is kind of oblong horizontally, I guess you could say, and then a far-sighted eye is oblong vertically. Some words to write down to go with this. Nearsightedness, your objects are more clear because the lens, here's the point. Here it is in words. The lens focuses on the image at a point in front of the retina. So the way it is shaped causes the lens to kind of make the light stop, kind of, before the retina, causing only the near things to be clear. In farsightedness, the far objects are more clear because the lens is focusing on the image at a point behind the retina. So the light is coming in, hitting back here, where it really, for good, perfect vision, if you will, it should hit right on the retina. So if it's hitting far behind it, you are farsighted. If the lens is focusing in front of it or near the lens, you are nearsighted. Please write down the scientific names as well in your notes. Myopia, I think is how you say that, and hyperopia. That's not how you say that. I've never had glasses. I need to look up how to say these words properly. Just write them down. Okay, okay. And then last slide is two other things that can happen to our vision. If we have damage to our occipital lobe, so meaning our eyeballs are just fine, our corneas aren't scratched, our lenses focus where they should, we might still end up with some unusual brain damage and or just genetic what would we call it? I don't know, areas of the brain that don't function properly. And we could either have total blind sight, which is exactly like it sounds. You could be totally blind, or you could have prosopagnosia. How's that? That is face blindness. We'll watch a little bit of a YouTube video of what that might be like in real life. But basically, it means that someone cannot register the image of a face. They can register surrounding hair, physical features, smell, body language, how recognizing someone based on how they walk, but they don't recognize a face, including their own. And that is exactly my 20-minute mark. So I'm going to leave you in suspense. We got some good demos coming up with this vision lecture. You're going to like the class that follows this lecture. All right, see you next time.